All right. Good afternoon, everybody. I'm just going to chit chat for just a second and give people a chance to filter in. See how we're doing. For those who are in already, as you're getting settled in, welcome and thank you so much for joining the conference and joining me here today. I see people are still trickling in, so I'll keep talking, and so people know they're in the right place, but we won't start until everybody's in. All right, we seem to be stabilizing. Okay, great. All right, well, welcome everybody. This is Formalizing Listening to the Land. I'm so glad to have you here. So I'm just going to share my screen. All right, there we go. All right, so welcome to Formalizing Listening to the Land, the importance of ecological monitoring. My name is Elijah Goodwin. I'm the Ecological Database Coordinator here at Stone Barns. And rather than leaving it till the end, when I have the chance of forgetting or running out of time, I just want to quickly acknowledge, you'll notice on the byline, this also says the Ecological Monitoring Team. I want to acknowledge that a lot of the stuff I'm going to be talking about today and some of the data I'm going to be showing is due to the hard work of a lot of people, including the people who kind of came before me and have been building and pushing for this ecological monitoring program here at Stone Barns, in particular, Jack and Shannon Algier and Shane Hardy. So I just want to recognize, plus a whole bunch of really wonderful interns, as well as the livestock team and the ecology team who've been collecting data through our GIS system. So thank you to all of them. All right, so let's get into it. Listening to the land is obviously not a new concept. So the tomb painting I'm showing here is from an Egyptian tomb from about 3,200 years ago of a farmer plowing with horned cattle. And fully recognized agriculture has been around for at least 10 to 15,000 years before present. And, you know, we can argue that number and what constitutes formal agriculture, but the point is agriculture has been around for a long time. And as long as there's been agriculture, and probably long before that, because even before agriculture, you know, nomadic hunter-gatherer societies were still altering the environment to improve conditions for game populations and for catching game, and improving conditions for gathering products of the forest and the grassland and so on. So, you know, listening to the land has been around for a long time. And if you didn't listen to the land, you were likely to go hungry or possibly even starve. So it's been an important part of human history for a long time. And if anything, kind of with modern industrial agriculture, we sort of removed recently some of that listening to the land. And you know, when the soil gets exhausted, instead of listening to it and giving it a rest or adjusting your agricultural practices, you just dump a bunch of synthetic fertilizer on it and keep going. So, but good managers and good farmers and land managers have listened to the land for a long time. So but that listening was at an informal level rather than a formal level. And what are some of the pitfalls of informal listening? Well, they're susceptible to bias. And so this diagram I'm showing here is kind of the classic cognitive bias diagram. So you tend to pick out the facts that match your beliefs. So when you believe that something is true, you tend to find the things that help support that and ignore the things that don't. And that's not always a conscious, it can be a conscious process, but it's not always a conscious process. We kind of, do that. And even if we see things that counteract over time, we kind of filter out the positive and keep the positive stuff and forget about the negative. We're also, as humans, we are designed to find patterns. And so it's very easy for us to find patterns where actually there aren't any. All you have to do is, take an image that has no faces in it and flip it over and join the two ends together and get that bilateral symmetry. And suddenly everybody's seeing faces if there's a complex pattern, because we are trained, our brain is trained to pick out patterns. So we can pick out patterns that aren't necessarily there. The impact of experience. So informal listening to the land really depends on your experience. So a new farmer who doesn't have a lot of experience may not be able to listen to the land in the same way as a farmer who's been farming for 50 years on the same piece of land and knows that land like the back of their hand. That you get a very different informal listening process from those different experience levels. Another thing is seeing the unseen. So there are things we can observe informally pretty well, plants, birds, animals, but there are other things like bacteria and soil chemistry and water chemistry that are really hard to observe without testing for them and without using specialized tools. So while we may focus on some of the megafauna and think that things are okay, we're completely ignoring a major part of the biodiversity on our fields. And with informal listening, it's difficult to change minds or attract investment. So if we want to tell people that regenerative agriculture is the way to go and they should be giving us ecosystem services credits, or they should be enacting policies that support and subsidize regenerative farming, we can't get people to do that unless we can show them the first thing they're going to ask us for is proof that these practices work. So we need some way to support our beliefs and present them to folks and help change their beliefs and their actions. Now, I want to make sure you know that does not mean that I'm saying informal listening has no value. It has great value. And that experience of that old farmer on the farm and what they know just by walking the land and feeling the soil under their feet and hearing the birds and hearing the insects, that's absolutely invaluable. The formal listening just gives us another tool to add to that arsenal. So why might we add formal listening? One is better management feedback. So again, because we are subject to bias and all those reasons I talked about in informal learning, plus the action of memory over time, sometimes it's hard to say, okay, is this practice working? I've done this for several years and I can't remember what it was like the first year. And so getting that formal listening, formal recorded listening to the land gives us better management feedback. So we know whether our practices are working or not. It gives us standardized temporal comparisons. So we can compare between years because we've standardized the sampling methodology. For instance, if one year we sample our carbon up on top of the hill, and then the next year we sample our carbon down in the valley at the bottom of the field where all the organic runoff is going, it's going to be hard to compare those 2 years and say, oh, we gained a lot of carbon because we didn't standardize the way we were looking at what was going on in the field. Formal listening, as I had mentioned in the previous, is very important for reporting to stakeholders. So how do we convince stakeholders that things are working, that we're doing the right thing, that they should support this work? And related to that, a specialized form of that is things like applying for carbon or environmental services credits. Again, we have to have the data to be able to show people that they're getting what they're paying for. It also can be super useful for public outreach and education. So again, how can we better tell the story to the public and better tell the story in our schools and to our visiting children about what is going on in the land and how this work is affecting the land. Again, a sub-aspect of reporting to stakeholders affecting policy. So again, if we have the data to back up what we're saying, then we can take that to lawmakers and make changes in policy. And it also allows us, for those of us who are interested, to contribute to or conduct research. And you'll notice on the conduct on the slide, I put a little asterisk. And the reason is that we can most definitely conduct research on our own lands, but we just need to be careful about how wide we apply those conclusions from that research on our land. Because, for instance, here at Stone Barns, we have fields spread out across quite a large acreage. You're actually looking at an image of some of our grassland fields right now where we graze livestock. And the problem is that a lot of those fields are geographically close to each other. And just because we put a road or a stone wall between those fields doesn't mean that that's a hard border that nothing can cross. These fields share soil types. The birds fly back and forth between the fields. The insects fly back and crawl back and forth between the fields. They get exposed to the same weather conditions. The livestock that we're running through these pastures are the same livestock, and often they're also carrying things from pasture to pasture in their hooves. So to treat each of those pastures as an independent point for scientific research would be a mistake. So we have it, to say this practice works all the time. If you're just comparing fields that are correlated with each other, dilutes the power, takes away the power of, dilutes the power of our ability to say anything with confidence. And by the way, if you have questions, please pop them in the chat. I'm going to try to end about 5 minutes, maybe even 10 minutes early so I can answer questions. But do me a favor, if you are going to be participating in my deep dive, if you could just put DD in front of your question so I can try to answer questions for folks who aren't going to be in the deep dive first, and then we can always go back, write those questions down, and we can always go back to them in the deep dive if we need to. So let's talk about starting or revising an ecological monitoring program. And we have recently revised our ecological monitoring program here at Stone Barns in collaboration with Terra Genesis International, who particularly Russ and Tim, who have been instrumental in helping us really do a deep dive into our ecological monitoring program, how we're sampling, how we're thinking about the big questions, and then how to collect that data and get that data into our system for analysis. So the first thing when you're setting up an environmental monitoring program or ecological monitoring program is you need to identify your stakeholders and what the big questions are for those stakeholders. And the stakeholders can include yourself as well. Again, your own management practices is a significant stakeholder for many folks. So whose needs do you need to serve? What information do they most need to know? How might you start to gather that information, and how will that information be communicated? So you're looking here at a diagram of all the different partnerships and collaborators, or not even all of them, but many of the different partnerships and collaborators that are part of our grassland ecological work. So in blue is Stone Barn Center, and we're in very close collaboration with in green Rockefeller State Park Preserve, who's really our partner in this work, because we are doing a lot of our grazing and our ecological work on park land in a cooperative management agreement called the CAP, which if you were able to attend Jack's session, he talked about quite a bit. And I believe Shane and Mike also mentioned that. So Rockefeller State Park Preserve is one of our big stakeholders. And so, for example, one of the things that the park is very concerned about as a stakeholder is grassland obligate bird populations. And they want to know how the grassland, they have a goal of bringing back grassland obligate nesting birds, and they want to know how those populations are doing and that those populations are growing. So we need to be able to design our ecological monitoring so we're collecting that information and collecting it in a way that is useful and that can be communicated to the park. And there may be different needs for some of our other stakeholders, like, for instance, Quick Carbon, who we've been working with on the Quick carbon testing. They need data from us to show that, to be able to show that they provide data to us, but we're also providing feedback to them on how well their tool is working and how it is correlating with the laboratory carbon testing that we're doing offsite. So the next thing to consider is, what specifically are you going to measure? So what are your monitoring targets and why? How will these targets address the needs of your stakeholders? And what specific aspects of those targets are you interested in, and how might you measure that aspect? So you're looking here at a little kind of ecological web I put together of the various ecological indicators that we're studying here at Stone Barns and how they relate to each other. Now, not everyone has the ability to hit all of these different targets. And so you have to decide what targets, given your resources, what targets are most important to you and most important to your stakeholders to work with. And so that will help you decide where to put those limited resources and what targets to focus on. You shouldn't start an environmental monitoring program by saying, this would be a fun thing to monitor, or, that might be an easy thing to monitor. Because if you're monitoring that thing and it's not meeting your needs and it's not meeting the needs of your stakeholders, it's going to be wasted time and resources. So for instance, if you had very limited resources, you may look at this and say, okay, one obvious thing might be soil condition. And you'll notice on this graph that soil conditions is actually over to the side and plant is in the middle. And plants just happen to have more connections. So when I structured this diagram, putting plants in the middle made the diagram come out well. But the fact that there's so many connections may lead you to say, well, plants depend on and affect so many of these other things that maybe that's the right target. And even if my stakeholders are not interested specifically in plants, I can make the case that by monitoring plants, we are getting good information on these other things. But of course, soil condition is one of the if I drew this as a pyramid, soil conditions would obviously be at the base. And so that's always a good target, basic target to get started with an ecological monitoring, particularly because it has such an effect on agriculture and is often something you're very concerned about on the agricultural end and not just the ecological end. So then you need to decide on your sampling methodology. So some considerations are sample size, and sample size also affects a whole bunch of different things: your cost, your time needed, your resources needed in people resources and equipment resources. And that sample size, again, should be meeting your needs. So if your needs are to know what's going on your land and know how some management actions you're taking, there should be enough sampling points to give you confidence that you know what's going on. If, however, you want to contribute your data to a scientific study, that scientific study may have a much larger sample size requirement to make it useful for them. Then you need to look at your sampling distribution. So there's different sampling distributions, how you lay out your sampling points across your landscape. And you can do that in a completely random way, and that has a lot of statistical validity. However, for many of us, a random stratified system is a much better way to go, because then you're making sure that you have points across your landscape, and each different management unit contains some points versus just a random where you may not get the coverage that you need for some of your other uses. And then there's also, you can also pick a fixed transect and say, okay, this area is representative of this field, and this is where we're going to do our sampling. Or this is the area that I'm particularly interested because this used to be an old hay field, and I want to see what's going to happen as we apply these practices to an old hay field. So, and that's a valid way to go, but that does reduce the ability for that data to be used in a statistical analysis. So it all depends on what your goals are and what the goals are of your stakeholders. What you're looking at on this map from our ArcGIS system is our quick carbon sampling points spread across the landscape. So you can see we used a program to randomly stratify those points across the fields that we were interested in sampling. So it's a random distribution, but almost, all the fields that we wanted points in have at least 1 or 2 points in it. And, the points are kind of allocated by the size of the field to some extent. So once you have your sampling methodology, how you're going to sample, and that can be different for different tests. So our sampling methodology for quick carbon is different from our sampling methodology for water quality, for instance, where we are targeting specific locations where we know it's a collection point from water from a certain point in the field. So then you need to look at detailed tools and specific procedures. So how are you going to measure? When are you going to measure? What is the sample handling in the field? And we'll talk about that more. And then you need to choose the tools and the methodology wisely. So there are a lot of tools out there. There are a lot of different methods. And again, you need to choose a tool and a methodology that satisfies your needs and the needs of your stakeholders, and also one that works with your environment. So you might have, you might want to use a, factory powered soil core to punch down a soil core down to 1 meter. And we'd love to use that here at Stone Barns. But the problem is our soil is too rocky. So if we went and invested in one of those, we'd find there would be very few, if any, locations on our farm where we actually could use that tool to sample, to do a deep carbon sampling. Then you need to match your methodology to your questions. Make sure you've written out your big research questions ahead of time, and now you need to know that that particular methodology will give you the answer that you need to answer those questions. That'll give you the right data. And then again, if you're hoping to contribute your data to research, scientists will tell you that if you're collaborating with scientists, they'll have a lot of requirements about how the sampling methodology is done and randomization of the samples and so on. But again, there's an asterisk, and there's one important and perhaps encouraging caveat there. And that is, again, that idea that all these fields are correlated. So really, in a study where we want to say, what are the effects of regenerative agriculture? Stone barns can really be only one point in that study. Now, by pooling all our data, for a particular management technique, that could be a very powerful point. It could be a very reliable point, but it's still only one point, because again, if we treat each of those fields as separate points when they're not really separate from each other, we're invalidating our statistical tests, and we're creating an error, and we're exceeding the inference space of our observation. So we can make inferences within our farm, but if we then try to make inferences to all farms, that's where we run into trouble. And related to that, a mantra for me is, don't let the perfect be the enemy of the good. Because if all you have bandwidth for on your farm is to sample one point, and you sample that one point well, that is better than not sampling at all. So don't get discouraged, and don't think, my data could never be used for research. Any sampling you do, particularly if you think about it ahead of time and it's well designed, is going to be useful. So let's talk about a little case study here. So as I mentioned, one of the goals for Rockefeller State Park Preserve is grassland and grassland obligate birds. And that's part of our CAP, or Conservation Action Plan, agreement with Rockefeller State Park Preserve, is that they want to see increasing populations and returning populations of grassland nesting birds, ones that nest on the ground in the grassland only. So we had to decide, all right, how are we going to measure this for the park? And so we decided the methods, and they're also interested in birds in general on and around the grasslands. So we looked at, what methods are we going to use? All right, well, we're going to look at biodiversity, and we'll look at breeding activity. But then we have to start narrowing that down. How are we going to do that? Okay, for biodiversity, we're going to look at point counts. And then for breeding activity, we have to have a methodology. How are we going to study it? So for our point counts, we're going to use the Common Breeding Bird Survey behavioral classifications when we observe birds, to look for behavior that qualifies as probable or confirmed breeding evidence. We're going to search specifically for grassland obligate nesting birds. And is it great that the birds are here? Yes. But is that what we want to measure? No, because that doesn't necessarily mean they're successful. Do we want to look at, are they here and nesting? Even better, but still, we don't know for sure that they're being successful. So what we really want to look at is their productivity, because that's what the park is most interested in. Do we have a healthy population, and is that population having the capacity to grow? And so productivity has to be our specific target. And then we design our methods. So, as I do this grassland obligate monitoring, I can input data into a map that goes directly to the phones of our livestock team. And so they know I can designate an area as a nesting area for a particular group of grassland obligate birds and say, okay, could in your next rotation, could you leave this area for now as a buffer around a potential nest? And then once I find a nest, I can narrow that buffer down to right around the nest. So we know we won't trample the nest, but we can open up a little bit more. We have an immediate functional aspect to the monitoring as well. So another case study, setting yourself up for success. So I wanted to talk just briefly about our 2019 insect monitoring. So we started out that summer doing insect monitoring. And it was a little bit of unsuccessful, let's say. And that's not anyone who planned its fault. That's certainly not the interns. We had some really great insect interns that worked on it. It's not their fault. We had to learn by doing. But what we found is, well, so there's 91,000 species of insects known in the US right now, and there's probably almost as many undescribed. Now, not all of those occur in this area, but it gives you some idea. Unlike birds, where a decent scientist, a decent ornithologist, a decent birder can go out and ID birds by sound and sight and get pretty much all of the birds identified, insects are really hard to identify. And in some of them, you have to basically dissect them and look under a microscope to tell them apart. And so it takes specialized skills, and even the people who are specialized in taxonomic identification of insects, morphological identification of insects, they're specializing in one particular group, and they don't necessarily have the ID knowledge for a generalist insect study. So what we found is we put so many hours we set malaise traps and pit traps and so on out in the fields. We collected lots of insects. Our interns worked really hard at trying to ID those insects. But after many, many, many man hours, we ended up with, a small section of the sampling actually ID'd, and many of those IDs were to like a family level, which from a monitoring standpoint is actually not super useful. So we actually had to re-look at our insect program and how we were going to carry it out, because we do think insect monitoring is important. And so it's important to have realistic goals and expectations, and again, really plan out that monitoring ahead of time and know what it's going to take so you can use resources effectively. And make sure what you're getting is what you actually need. So you're getting species level identifications instead of family level identifications. So one way to do this is to utilize new methodology and technologies. So one thing we're looking at is working in association with the University of Guelph's Center for Biodiversity Genomics, and they have an insect DNA barcoding program. The great thing about this program is you have to set up a regional library first. And so there's an initial outlay, which we're working on getting funding for. But once we do that, you can submit bulk bottles. So basically you can submit a bottle full of insects. They'll grind it up, they'll run the DNA barcoding, and they'll be able to tell you what species are in there based on matching to the regional library and other sequences in their database. And they'll be able to tell you if there's something in there that they haven't seen before. And so it gives species level identification. And it is really, for what you're getting, it is really inexpensive. At $50 for like a bottle of 1,000 insects, you can get species level identification of most everything in that bottle. And that is a huge game changer. And so our hope is once we set up this regional library, we can give people access to that regional library, and we can pool sampling. So we can actually create a regional insect sampling service that allows us to ID insects much better. Just being cognizant of the time, we're at 10 minutes here, so I'm gonna hurry along. All right, so case study 3, where does your data go? This is another thing that we kind of learned the hard way. We had lots of interns on campus. Everyone was kind of responsible for their own data they were collecting. We had data in Google Docs here and there, Excel spreadsheets, etc. Some people had the data on their personal devices. And we didn't know necessarily where it was. It was hard to get access. It was hard to make the data talk to each other. So when you're setting up a program, you should really think about how you're going to record your data. How is it standardized, right? So if one person records the common names of the birds and the other person uses a banding code, at some point, to make those two databases talk to each other, you've got to translate from one to the other. So if you can put it in standardized in the first place in the field, then that data can talk to each other and be joined together very easily. Then you have to think about what other data products and physical samples are produced. So, are we collecting soil samples? Are we collecting archive samples? Are we collecting pictures, etc.? So what other things are going to be collected? Are we collecting insect samples to use as a reference library? And then we need to think about where are these things stored and how. We need to be able to store them in a way that preserves those for use later. One thing you might think of is, okay, let's say we wanted to store some insect samples in a preservative. Well, we may want to think down the road at some point, maybe we're not doing it now, but at some point we might want to use those archive samples for DNA analysis. So we do not want to preserve those insects in a preservative that's going to degrade the DNA for later analysis. So these are the things that if you think of ahead of time, it saves you heartache later on. And then how and when will they get accessed and analyzed? How do you get access to that data? Who can get access to that data? How can that data be condensed and analyzed and visualized? And so the slide here is showing part of our GIS system that we just created, where we have mobile apps, Survey 123 and collector apps through the ArcGIS system for our field workers to collect data in a standardized way in the field. That data goes directly to our database. As you know, if that cell phone has signal, instantaneously practically. And if it doesn't have signal, as soon as that field worker gets back to within Wi-Fi, all that data uploads to our system and it's ready to be synthesized, visualized, and used. All right, some notes, quick notes on monitoring tools and procedures. General sampling considerations. Think about the seasonality of sampling. If one of the things you want to monitor is spring ephemeral wildflowers, you need to make sure you're out there at the right time of year and you're monitoring, since those blooms can vary by several weeks. You need to make sure that someone's monitoring when the bloom is happening, and you need to be able to be out there with the appropriate number of folks you need to get that sampling done within the window that those flowers are out. Think about fluctuating environmental conditions. So one place this really comes into play is if you're doing water quality sampling. Studies have shown that the pulses of pollution, from pollution sources, both agriculture and otherwise, generally come during high flow events when there's a big rain. And it's some point after that high flow event when that surge actually gets to the point where the monitoring is done. And so if you're only going out and monitoring on good days, on sunny days, you may be getting a very different picture than what's actually going on when those pulses are coming through. You need to think about sample handling, storage, and shipping. So we're actually looking at one of our incredible interns, Emily, from this summer, who's doing soil sampling for the soil samples we send out to Woods End soil labs. And one of the tests there is aggregate soil stability. Well, we do a composite sample. So as we are taking those samples and mixing them up in the bucket, are we actually breaking down some of the aggregate stability that we want to be able to see? So sampling handling can be huge. Or if you're collecting something, soil for biological analysis, for biological activity analysis, or plants for nutrition analysis, and then you're throwing them in your truck and they bake in the hot sun for several hours, you are changing what you wanted to, the important data. And so you basically wasted all this time doing the sampling, and because you didn't treat the sample properly, you didn't get the data you needed from it. Field testing versus laboratory testing. So in some cases with things like aggregate stability, maybe it's better to do a field test that is not quite as fancy and elaborate as the laboratory testing, but because you can literally take that clod of dirt right in situ without compressing it, without breaking it up, without subjecting it to drying and tools and so on, you might get better data and a better picture of what's going on from that field testing versus the complicated laboratory sampling. Composite sampling is a way that you can get samples from over a more representative area of your field. So you take a bunch of samples and mix them together. An example would be, on our deep carbon transects, we take, for the soil sample, we take random samples around each of the deep carbon transects and then mix those together. Or for forage analysis, we take, you need like a pound of vegetation. So we take clippings from a variety of points around the field. All right, and we are running low on time, so let me head on. So sampling should match the needs. You should look at right tools for the conditions, as I mentioned. With that deep carbon sampling, if you have rocky soil, you cannot use a soil core. And you should moderate expectations. Don't base on 1 year's difference that something is working or not working. Environmental systems take time to adjust to new management schemes. Some quick review of sampling technologies. Quick carbon, that's a quick carbon reflectometer. So this is another way to be able to sample more cheaply, quicker, often in the field. Quick carbon uses a reflectometer to scan the soil, and once you've calibrated with quick carbon your soil profile, they can take those reflectometer readings that you get through your phone and give you a readout of the carbon content. Microbiometer is a card where you mix the soil, you place a dot on this card in the middle, and you take a picture with your phone, and it will, based on the spectrometer reading, it will give you a readout of how much microbial activity you have in your soil. Coliscan is a water sampling tool where you can quickly scan for coliform bacteria in your water. Water quality sondes avoid that problem of those pulses. You can leave a water quality sonde in the water source and get constant data monitoring. Drones allow us to do things like use an NDVI sensor and get data on photosynthesis in our fields from above. Remote sensing also, there's a lot of free remote sensing data that provides a lot of really useful data. GPS tracking for cattle is one way to see where your cattle are going in the field. And then again, we mentioned DNA technology with the insects. That's a rabbit hole that provides a lot of really useful tools. And it's not just the results. Record your management activities. And you can see here, if I click on this map, we're getting a readout of everything that's here. So we have a polygon in there for mowing and all the data for the mowing. for when it was tilled with a keyline plow, when there were chicken pastures there, when the mixed flock of cattle, sheep were there, when the weaning flirt was there, and so on. So you can see this gives you, if you want to see how your management actions are affecting the ecological variables, you have to record where the management actions are occurring. Finding partners, reach out to universities, colleges, schools, and nonprofits, reach out to local interest groups, bird clubs, photography clubs, reach out to adjacent public land agencies, create an ecological monitoring cooperative with like-minded farms, look for local volunteers and summer interns, whether college or high school, and don't be shy about reaching out to people, particularly if you have a local university or college. Reach out to them and say, if you have any students who are interested in doing research, my farm is always available. And then provide value for those interns and give good feedback and support. A couple of open source things: Open Team, FarmOS, QGIS, the R project, and NRCS. And if there's one take home from this today that I'd like everyone to get, is that everyone can do some ecological monitoring, and just that planning beforehand is crucial to being successful. So thank you very much, and I'm sorry we ran low on time for questions. Let me stop the share. And I see, is there any monitoring techniques for native pollinators in their life cycle? So that insect, the DNA barcoding could be great for pollinators. Some pollinators are a little easier to ID, some are still hard. So you could do a morphological monitoring or a photo monitoring program. There's also really cool stuff with eDNA. So you could actually sample from a flower and use eDNA, the traces of DNA the insects leave behind, to see what has visited that particular flower. Is there any way we can get a link to this presentation? Yes, I'm gonna post a link to the presentation in the chat right now, and I'll also make sure it gets up on the YFC site. So there we go, let's copy this. So this story map will remain public, and you can get access to it right there. And then you should be able to see the open source resources. All right, so we probably should wrap up. So thank you so much for coming, and I look forward to talking with folks in my deep dive. And if you have any questions, please reach out to me through the Socio app, and I'm happy to answer any questions you have. Thanks very much.